Welcome everybody to this first edition of the EASD Academy Journal Club in collaboration with Diabetologia. This activity aims to provide authors an early stage of their career with the opportunity of disseminate their findings and inspire other colleagues. For this, papers accepted for publication in Diabetologia will be selected by EASD Academy, and the first author of the paper will be invited to present and discuss the work in a live seminar like this one. So the webinar will include a questions and answers session when the audience will be able to interact directly with the speaker via chat, and we hope you enjoy this new experience with us. Soon we're gonna start with another activities and new webinars that will be advertised in our web page, so stay tuned. My name is Beatriz Merino. I'm basic researcher from the University of Valladolid in Spain, and I'm co-chairing this session with Dr. Ben Stokes, a basic researcher from the University of Copenhagen in Denmark. And we are members of the EASD Academy Committee, and we are happy to accompany you during the course of this session today. So for this first event, we are a pleasure to have here with us Samantha Yoon. She's first author of a recent paper in Diabetologia. And in this session, we will the opportunity to listen to Samantha's work, and then we will open the discussion. We would like to be this dynamic event, so we encourage you to ask any questions or comments in the chat, and we will be happy to pass them to our speaker. So first of all, I would like to introduce Samantha. Samantha is an early career basic researcher. She obtained her biology bachelor degree in 2016 in the University of Columbia. And nowadays she is PhD candidate in the laboratory of Dr. Francis Lin in the BC Children's Hospital Research Institute in Vancouver. And her recent article in Diabetologia is entitled Calcium-Dependent Transcriptional Changes in Human Pancreatic Islets Reveal Functional Diversity in Islet Subtypes. So thank you so much, Samantha, for accepting our invitation. You are welcome to introduce yourself in a little more detail and start with the presentation of your paper. And finally, I would like to ask to the audience to keep the microphones off in order to listen to Samantha properly. So thank you so much, and go ahead.
Okay. Well, first of all, thank you to the organizers for inviting me to become the first speaker for the EASD Academy Journal Club. You know, I've actually never even been to an EASD conference myself, so this is actually very exciting for me. And I'm looking forward to just quickly going over my recent work that was published in Diabetologia with you all, and hoping to get some good feedback out of it and some discussion at the end. So I've been designated about 10 minutes to go over this paper. So obviously I'm not going to be able to go through all of it, but I want to really focus on the overall story and how this project got set up, and some of the key findings that we published in the paper. And if anyone needs to know how to find the paper, it's I have the DOI posted on every slide. Before I just begin the presentation, just want to do a quick land acknowledgement that this is UBC campus, by the way, where the institute I work at is not located, unfortunately, because it's beautiful. But all the research that was performed at BCCHR as well as on UBC campus is located on the traditional, ancestral, and unceded territories of the Musqueam people. Okay. So in the Lin Lab, when I first joined, we mostly studied beta cell function with a focus on the transcriptional regulation in mature beta cells. So I've just plotted up a little diagram here. Most of you probably recognize this, but this is the classical model of glucose-stimulated insulin secretion in a beta cell. So we have glucose entering the cell through transporters, gets metabolized into ATP. And following the closure of the ATP-dependent potassium channels, there's a depolarization event that leads to a calcium influx through voltage-gated calcium channels. In granules, we know that in other islet cell types, this last step of calcium influx directly triggering the exocytosis is present as well. I just got a warning saying my internet connection's a little unstable, so I hope I'm not lagging out too much, but I'll continue. Yeah, and we also know that calcium, being a potent secondary messenger, can rapidly act on other processes within the cell. And one of them that we studied a lot in the lab is this process of calcium-regulated transcription. So focusing on the beta cell, which was the lab specialty, we know that this process of calcium-regulated transcription really heavily depends on calcium-binding proteins, such as calmodulin, that can then further activate kinase signaling cascades and other proteins such as calcineurin. And this eventually leads to the translocation of various calcium-sensitive transcription factors, essentially. Well-known ones include the CREB and NFATs, and they in turn can induce expression of certain response genes. And we really became interested in this process because at the time, the lab was studying one such response gene called NPAS4. And NPAS4, we found out through 2 PhD students before me that it was a very specifically and robustly calcium-induced gene within the beta cell. And it's a transcription factor that can then induce its own target gene expression. So we characterized it a little in the beta cell and found out that it was actually very important for beta cell function during high glucose stress. So that was work that was done kind of before me and when I first joined the lab. And at the time when I joined, this was now, I think 4 or 5 years ago, we were finishing up the NPAS4 characterization in the beta cell, and kind of naturally, we got to thinking, we should broaden this question a little bit more and start to think about, well, if NPAS4 is a calcium-regulated gene that's important for beta cell function, how many other genes are there that follow this kind of regulatory pattern? And if we expand this now onto the scale of the whole islet, how would certain gene sets differ downstream of calcium signaling in the different islet cell types? So really, through this project, the questions we wanted to specifically answer were, what are the genes that are regulated in response to specifically calcium signaling within the human islet cell types? And if there are differences either across cell types or within a cell type, would this indicate any functional differences aside from the transcriptional differences? And really, it was lucky for us that at the time, we were just starting to set up the 10x Genomics Chromium single-cell RNA sequencing kits within the Institute. So it's like I was there at the right place at the right time. The project was available. I was also interested in it. So that's the stars kind of aligned, let's say. And this project kind of took off. We started by obtaining islets from human cadaveric donors that were sent to us by the Alberta Islet Corps in Canada, so one province over. And then when the human islets arrived in the lab, we split them up into 3 different experimental conditions. We try to keep it as simple and robust as possible, hence the kind of super physiological stimulatory conditions you'll see here. So we placed the islets into, there was a low glucose condition that I just called low and the name stuck. There was a stimulatory condition of very high glucose and high KCl, and that double whammy was really to maximally induce calcium signaling within the islet cells. And I should note that all of these conditions were placed on intact islets, so they weren't dispersed at this point. And finally, to contrast against that stimulatory condition, we provided the islets with a condition that I called negative. It's the same high stimulatory condition, but with a EGTA calcium chelator added. So that's an extracellular calcium chelator that is and we added it in essentially to prevent that calcium influx from happening. You'll notice that we only place the islets in these conditions for 1 hour, which isn't a terribly long time. And this was really because we wanted to focus on the most rapidly regulated and the most robustly regulated calcium responsive genes. Following the 1 hour, then the islets were dispersed, and then we performed single-cell RNA sequencing. And I'll just kind of jump right ahead to the punchline, but as you can see here, so this is from figure 1 of the paper, but we did detect pretty much all the cell types that you would expect to be present in a human islet prep, of course, with a more heavy emphasis on the endocrine cells, because these were manually picked islets. So you'll see trace amounts of ductal and acinar cells, but mostly it's endocrine. And I really do have to state that I was not from a bioinformatics background of any sort when I began this project. The bulk of the analysis and the data handling and the data coding, setting up the pipeline for analysis back then, it was really like these 3 undergraduate students back then were very instrumental in this entire process. Okay, so a couple of, I'll start going through what I believe are the really important points of the paper. So the whole point behind why we did these 3 different conditions was so that we can start to compare the transcriptomes across donors between these 2 conditions, particularly the positive and the negative, so that we can start to pick up genes that are differentially regulated between the 2 conditions. So for example, if we look at what we hypothesize to be a classic expression pattern of a calcium-dependent gene, we would see a big difference between the gene's expression under the positive condition versus the negative condition. So we essentially performed this comparison in every single cell type and every single cluster within that cell type across all the endocrine cell populations to begin with. And we found essentially calcium-regulated genes that were shared or specific for a certain cell population. So you'll see that delta cells don't actually express a lot of unique calcium-regulated genes. Alpha and beta cells do share some overlap, but mostly they have their own separate gene sets. And it gave us great happiness to see that the gene that we had already confirmed to be calcium regulated and PAS4 did show up in the beta cell population. So that was a good positive control almost for what we expected. I'm not going to talk about any gene in specific. I actually want to focus more on the latter part of the paper, which kind of got started through a little bit of a different way of thinking. So normally what we would have actually done is, I think, go after the genes individually and confirm them and validate them. Instead, what we wanted to focus on are the overall numbers of the calcium regulated genes. And we interpreted that as the essentially magnitude of response to calcium. So if we focus on, we identified 5 different beta cell clusters within our beta cell population in the data, but we really focused on beta 3 and beta 1, which were the 2 clusters that expressed and regulated the highest numbers of the calcium regulated genes. And when we compared these 2 clusters against the rest of the beta cells, essentially, we found that one of the genes that was most enriched in these 2 clusters was a gene called PCDH7. You can see that it is fairly specific for the beta cell population in our data. It is particularly enriched in the beta 3 and beta 1 clusters. And it was kind of lucky that PCDH7 does encode for a protein. It's a protocoherine encoding gene, so it's found on the cell surface. And Dr. Shugo Sasaki, a postdoc in the lab back then, he jumped into this project at this point and said, well, if it's on the cell surface, we can sort for it with an antibody. So he did that. We went and obtained some more human islets, dissociated them, and sorted out PCDH7 protein expressing cells and cells that did not. We re-aggregated them in agar-wool plates that our lab typically uses for stem cell culture, but they also worked in this case to re-aggregate essentially PCDH7 positive and negative cells into almost like pseudo-islet clusters. And then he performed a static glucose-stimulated insulin secretion assay. And we're using human C-peptide as a surrogate measurement of insulin here. But as you can see, the cells that express PCDH7 on their surface respond a lot more to the glucose stimulation and also the depolarization by KCl. And there's calculated stimulation index is a lot higher than cells that do not express PCDH7. So then we moved on to actually characterizing these cells a little further. We were worried that the enhanced function could have been due to a difference in some sort of maturation state. These were from adult donors, but we examined the expression of certain key beta cell maturity genes, and we found out that the enhanced function was not due to any differences in maturation state as far as we can tell. So we are currently pursuing with the thought that PCDH7 is a marker of beta cells with enhanced, essentially calcium-regulated transcriptomic responses and also increased glucose-stimulated insulin secretion. There is one other finding that I want to quickly touch on before I wrap up my presentation, and that's the presence of these kind of dual hormonal, bihormonal insulin and glucagon expressing cells within our data. They are pretty rare. They're about 3% of endocrine cells. So if you take it on the scale of a human islet, it's probably less than 3%. But they express both insulin and glucagon transcript. And at the time when one of the reviewers actually really wanted us to go back into human islet samples and pancreatic sections, and essentially they wanted us to show that these cells existed within the native human islet. So many sections later, we did that. So these are either embedded islet sections or human pancreas biopsy sections. And these are not immunostained images, I should clarify. This is performed with a kit that's commercially available called RNAscope. It's a single molecule FISH kit. So this is directly detecting mRNA molecules. So you'll see the insulin green here and the glucagon in red, but across multiple donors, and especially with the pancreas biopsy sections, we actually went back to the donors we used for the single cell experiment. But across multiple donors, we do see presence of cells that co-express insulin and glucagon transcript within the same cell. And you can see the co-localization here with the arrowheads in yellow. So I'll just quickly summarize, but essentially what we did was we started with a very simple question, and we performed single-cell RNA sequencing on human islets exposed to multiple stimulatory conditions. And this was all to determine the specific calcium responsive gene sets that are alpha, beta, and delta cell specific. And furthermore, we kind of use those calcium responsive genes, at least the absolute number of them, to use that as a surrogate measurement almost of the calcium responsiveness of a cell. We found that beta cells that regulate the most calcium responsive genes are marked by this gene PCDH7. And at the protein level, PCDH7 positive cells do show enhanced insulin secretion over PCDH7 negative cells. And then within the paper, I think I go into a little bit more in depth about this, but we did find these rare insulin glucagon cells that do regulate their own set of calcium responsive genes. And some of them are distinct from alpha or beta cells. And we did do a little bit of work in the paper to show that they might be a little less mature in identity, but compared to alpha and beta cells at least, but they're not, we don't think they're actually sequencing artifacts or the product of perhaps dedifferentiation or transdifferentiation, but we actually don't know a lot about these cells so far, but we did go back to islets, and it seems like they are a real population. So that was good enough for us, honestly. So I would like to finish by saying, obviously none of this work would have been possible without my supervisor, Dr. Francis Lin, who's here, and the rest of the Lin Lab members, both current and past, especially the people highlighted in bold here. They were really instrumental to getting this project started and finished. Of course, funding this wouldn't have been possible without the support of CIHR and Canucks for Kids Fund. And finally, the ADI Islet Corps for sending us the samples. And obviously, big thank you to the human islet donors and their families for making this research possible. So I think, yeah, that's it for my presentation. I believe we can move on to questions now.
Okay, thank you so much, Samantha. Your presentation is really nice. Your work is impressive. And first of all, before starting with the science questions, we would like to ask you a little bit about your career and your involvement in the project, especially for the inspiration of the young people. And in this regard, the question is, what has this work meant in the development of your career? Are there any turning points that you can share?
I mean, honestly, this entire project was a little bit of a turning point for me, and I'll explain what that means. So this is obviously an islet paper, and the Lin Lab is a beta cell lab at its core. Like, a lot of people study beta cells, stem cell derived beta cells, but I actually am focusing on neurons as the bulk of my thesis project. I study activity regulated genes in hypothalamic neurons. So that's the project I actually started my degree with. And right around in my third year, Francis called me into his office and said, do you want to, we're trying to get single cell set up in the lab and we want to start a human islet project on them. It's a little removed from the other work you're doing, but maybe do you want to give this project a try? And at the time, there were, I think, several senior students that he could have asked to take on this project, but for some reason he asked me, and for some reason I said yes. He did say that he thought it would be a quick and easy project that would yield a paper in 2 to 3 years. Maybe that was the selling point. So what did I know back then, honestly? But I think just agreeing to kind of step outside my comfort zone and tackle this, because we also didn't have anyone who was particularly experienced with bioinformatics or omics data at the lab at that point. So collectively, I think it was a big challenge for the lab. And at the end of it, I am kind of happy that I agreed to take this on. Yeah, that's the best answer I can give you at this point. So.
And how about the funding of this project? Have you been personally involved in get funding of these projects? And you are applying for grants or awards or something like that?
No. So this project, the funding for this project came from CIHR, which is the Canadian National Funding Agency for Science, one of them at least. And by my understanding, I think this is more outlined in the acknowledgement section of the paper, but my understanding is that the funding from this came from a grant that my PI had already obtained for a number of years. And it was actually before I even joined the lab. So I was definitely not involved in any portion of the grant writing process. And even in the lab, I don't think Francis actually, I think he prefers writing grants on his own. I know he asks the postdocs in our lab to give him figures and data sometimes, but for grad students, we're typically not involved in the process of grant writing.
Okay, so we can move on the scientific questions and technical question of your work. And I have 2 little questions, and then we can move to the questions and answers from the audience in the chat. And my first answer is that the islet that you use were from male donors. And you know that sex differences in glucose homeostasis have been reported in another publication. So the question is, are you aware of sex differences in calcium regulate genes? And do you think that this could be worth exploring as a mechanism responsible in sex differences in glucose homeostasis?
Yes, like, do I think it would be worth exploring? Absolutely. It's actually one of the, I guess, like, I like my paper, but I also have a lot of regrets about the paper. And a lot of that, I think, is to do with the limitations of the particular donors we use. So you're absolutely correct. And the donors that we used were all male. Actually, in the earlier stages of the project, we actually did perform the same experiment for a female donor's islets. And we just never got around to incorporating that data in the final version of the data set. One, because we were unable to find female donors that matched the age and BMI and HbA1c of the first female donor we sequenced. And second, because we had a discussion about it and we unfortunately were limited by just the amount of money that we could actually spend into this project. So exploring both sexes, I think, would be an absolutely crucial thing to do in the future as a kind of a continuation of this project. I'm not sure if I'll be able to do it, but it wouldn't surprise me, honestly, if we repeated it for a female donor set and there were either different calcium regulated genes or there were different numbers of calcium regulated genes. Or I could be completely wrong. There could just be different subpopulations of beta cells altogether if we compared male versus female. So I think sex differences are important. So it's one of the bigger regrets of this project that I wasn't able to fully pursue that just because of physical limitations.
Okay, and another question. You demonstrate that protocadherin 7 mRNA is present in beta cell and that expresses higher number of calcium regulated genes. The question is, do you think that protocadherin 7 could be a biomarker? And if you can tell us something more about the protocadherin 7?
Yeah, so PCDH7, whether it could be a biomarker, I mean, it's not secreted, so I'm not really sure how it would become a biomarker that you could detect for in humans. So no, I'm not really sure at this point how it could become a biomarker, but I can tell you a little bit more about PCDH7. So the thing is, we don't really know a lot about PCDH7 beyond the fact that it is a protocadherin. The proteins expressed on the cell surface, about 20% of beta cells seem to express this. But whether it's mechanistically involved somehow in these higher calcium responsive cells, we actually don't know that. We're interested in following up on it. We know that I think in mouse islets, PCDH7 is actually, like, the expression is either very low or undetectable. We were actually trying to find maybe like a floxed mouse for it and see if we can generate a knockout model for it to follow up on it, but that wasn't possible. So something that we've actually considered doing, and again, I don't know whether I'll be doing this or not, but the rest of the Lin Lab actually focuses on stem cell derived beta cells. So we're primarily a stem cell lab now. And what we've actually performed single cell RNA sequencing on several of these stem cell derived beta cell spheroids. And we actually see PCDH7 expression in those spheroids overlap with the cells that have the highest expression levels of genes that you would expect in a mature beta cell. So these are the insulin expressing cells in the data set, because the spheroids end up kind of being a mismatch of mature and immature cells. So we think that maybe studying PCDH7 further in these stem cell derived beta cells could potentially give us more of a clue to its actual role, whether it's mechanistic or whether it's simply a marker. And if it is a marker, when does it turn on really during development and maturation of these islets?
And in this line, what will be the take home message of your project?
Oh, so many, but if I had to pick one, well, the scientific take-home message, I think, is that single cell really depends on the samples you start with. So I think a lot of people, whenever I've presented this work in the past, a lot of people like to ask me like, oh, but what does this mean for all these other donor profiles out there? I don't know. Our data set was very limited to 3 donors. They were males in their 50s. They were non-diabetic. And really that's the donor pool that our data set is limited to. So beyond that, I don't know how much of our findings we can try to extrapolate and find in other donor profiles. So the take-home message would be like, you know, I liked how this project turned out, but I don't think we should ever fool ourselves into thinking that one single cell experiment is going to yield the results for all of the samples available in the donor pool. So I think that's why validation is really important. Multiple data sets generated by multiple groups across the world are important. So I'm not saying everyone should be doing single cell. I think it's a technology and I think it's a useful tool, but I think it also, because it has so many moving parts, it requires a little bit of caution, especially nowadays when there's so many tools available to analyze and deal with this kind of data.
Great. So thanks, Samantha. And I think that is time for move to the questions and answers of the audience. So Ben is opening up this part.
Yes, thanks, Bea. Yeah, so we've already got some questions in the chat. So Rodrigo, why don't I invite you to unmute yourself and ask a question directly?
Hey Samantha, thank you for the nice presentation. I was just wondering if the gene that you find, I mean the name is you know, much better than me, PCDH7, maybe it's also differential expressed in type 2 diabetic islets, or if there is any known SNPs in this locus that is associated with type 2 diabetes race?
Yeah, that's a good question. We've not looked at either, to be honest, but we do have, like, neither of those questions would be particularly difficult for us to look at. We do have perhaps type 2 diabetic donors saved up, although they're a little bit harder to come by, I think, overall. So I think, no, the SNP one I can't answer. I think I did actually try searching up whether there were in a publicly available one of the GWAS databases. I think I did try searching up for PCD7. I did not find any, but this was several years ago, so I should probably look again. Thank you for the reminder. I'll need that for my thesis.
And I don't know, Ben, if I have the time to ask the second question, or
Yeah, ask the second one, and I will invite anyone that wants to ask a question to either raise their hand or put a question in the chat. But yeah, go ahead, Rodrigo.
Thank you. So the technical question is why you stimulate the islets with glucose plus high KCl and not only high KCl, considering that glucose also will, of course, like be metabolized and make so many change, and also will foster the transference of calcium from ER to mitochondria and mitochondria uptake of cytosolic calcium. So it will be many change of calcium inside cells. So and somehow could be a confounding factors as well. I don't know.
Yeah. Oh, definitely. The conditions that we expose the islets to, yeah, it's essentially the equivalent of hitting them with a sledgehammer, right? It's a very, very strong, super physiological level of glucose that probably a non-diabetic islet has never encountered in its entire lifespan. And KCL, in addition to that, like we're definitely aware that it's a very strong stimulus. It is, however, simulatory conditions that we've previously used in the lab back when we were characterizing NPAS4. So we did feel a little bit of justification in using it because with our reasoning was that if we don't see anything when we hit the cells with a sledgehammer, we're definitely not going to see anything when we lower the concentration and start following up on any hits we find. So that was one. We essentially wanted to push the islets to their maximum a little bit. And yeah, I think that's the best response I can give at the moment. It's not the best answer, but it's a realistic answer.
Thank you, Samantha.
Cheers. I'll ask a question in the meantime while people think of their own. I do a fair bit of omics work, and yeah, you get a lot of hits, right? And then the difficulty to make an interesting paper is to choose what to follow up on, right? And so why PCDH7? What's jumped out about it in your data set to go, right, that's the one that we need to follow up on?
Yeah. Well, the PCDH7 hit, I guess, occurred a little bit. Actually, it occurred right after everything shut down because of the COVID pandemic. We had a lot of free time to work on this data set from home because that was, for a while, the only work that we could do because we couldn't go into the lab. So we spent a little bit of time on it, and because the list for calcium regulated genes that's a separate list from the list of genes that were, I guess, enriched for those calcium responsive clusters. So you're right, this was a much longer list. PCDH7 jumped out at us because, one, it was near the top in terms of log fold changes, and also p-value, but I don't really, like, when it comes to single cell, p-values aren't really, you know, they're not the best measurement of significance, I think. The second thing that PCDH7 kind of prompted us to follow up on was because it had actually been, it wasn't studied specifically in islets before, but there was a paper, I'm forgetting the year, but essentially the paper's title was something like there are 4 subtypes of beta cells. I think the author's name was Grompe, but if I recall correctly, it was like CD9 and another cell surface marker. And using the combinations of these 2 cell surface markers, the authors essentially said there are 4 subtypes of beta cells. One of the subtypes that they showed, and they did RNA sequencing on these sorted subtypes of beta cells. One of the subtypes actually was also enriched for PCDH7. And that subtype in that paper, those beta cells also, they also showed that they had an increased Jesus response. So when PCDH7 popped up on our enriched gene list, and we're going down the gene list, and we're kind of searching up, like, well, we don't know this gene, what's known about this gene? It's just one of the hits that happened it linked us to that paper, and we figured, like, oh, that's interesting. Well, what if it's not the cell surface markers that they're talking about, but what if it's this other one instead? So that's serendipitously, that's how we kind of ended up following up on PCD7. So as with all omics data, it's a mix of luck and a little bit of Google.
Absolutely. Hendrix put a question in the chat. So if you would choose, how would you validate your findings and find the role of PCHD7? Yeah, Hendrix in the car, so I'm asking on his behalf.
Well, I pitched this idea to Francis, and he seems to be okay with it, so I guess we'll end up doing this. But I wanted to, well, first I wanted to go to the stem cell derived beta cell line that we have in the lab, and I wanted to just simply start by sorting out, in the same way I did in the paper, sorting out PCD7 expressing cells, see what's different about those cells versus the other stem cell derived beta cells. But I also wanted to try overexpressing PCDH7, or the opposite, I guess, knocking it down in some way, and then seeing how that affects the activity of these stem cell derived beta cells. So presumably, if PCDH7 played an actual mechanistic role somehow, that caused these cells to become more calcium responsive as they develop, and maybe that persists onto adulthood. We would see that we would see a difference between the cells that overexpress PCDH7 versus cells that didn't, or cells that had knocked down PCDH7 versus the ones that retained normal expression. So I don't know if I will have time to do this before I defend, but that is kind of a line that we are following up on.
Nice. And maybe I'll ask another question. So you spoke a little bit about these insulin and glucagon cells, the like a small percentage. And can you talk a bit more about those and potentially what the significance of that finding may be to metabolism or beta cell function or whatever?
Yeah, I mean, we don't really know much about these cells apart from the fact that they're rare. And like single cell is always a snapshot in time, right? So for all we know, this could just be some sort of very transient intermediate cell state, not necessarily a defined and persisting cell population. What we do know is that they seem to be present in very small numbers, at least in the islet sections that we've looked at. And we did, I think it's in like figure 5 of the paper. We did attempt to run some velocity analysis on it, and like no one's an expert in velocity or pseudotime in the lab currently. But our interpretation of it was that it didn't seem to be the result of any de-differentiating cell from a mature cell state, or like some sort of trans-differentiating cell state, but it didn't seem to be some sort of immature pool that gives rise to other mature cell states either. So we're not really entirely sure what this means, but I kind of like to think about it like this. Like, I think what you find at the transcript level and what you find at the protein level are entirely different worlds. We don't know if these cells actually secrete insulin and glucagon. What I do know is that I think I went to a conference in May of this year, and after I talked about these cells a little, other people started coming up to me and say, hey, we also see these double positive cells in our prep. So presumably, if we're all seeing the same thing across the globe, then at the very least, they must be real. Not sure if it carries over to the protein level. But I think it just leaves kind of this fun extra kind of avenue to explore later on down the road. I think there's a lot we don't know about islets still, so that makes it kind of fun.
And were you able to find a marker protein for these?
No, that's what's so infuriating. Yeah. Like, that was the first thing I did, right? I was like, we got to go after these. We got to see if they're real. Yeah, unfortunately, they don't really seem to express particularly enriched levels of any gene that codes for anything on the cell surface. What I do recall is that they did express kind of unusually high levels of genes that code for ribosome subunits. So perhaps there's a lot of translation happening in those cells for some reason, but yeah, apart from that, we didn't really have a lot to go after. We tried looking at all the canonical alpha cell markers, beta cell markers, just kind of expressed middling levels of all of them. It was very infuriating. Oh, I see a question in the side. Do they express markers of immaturity? That's a difficult one. If you're asking whether they express, like I checked for markers of endocrine progenitors. Yeah, they didn't seem to have those. But when you compare a list of, and I think this is also in the paper, but it might be in the supplementals. But we did compare the expression levels in insulin glucagon cells versus alpha cells and beta cells of just well-known beta and alpha cell maturity markers. Again, just lower levels of all of those, but nothing that was particularly standing out to us.
Yeah, have you tried silencing the gene to see if it directly affects insulin secretion?
Yeah, that would be, I think if we do end up at the point, well, I feel like he's asking if I've tried silencing the gene in mature islets, and no, we have not. We haven't done that. But that's a good idea. Maybe we can try that. Yeah, we can definitely try that for sure. And then that in combination with like some sort of knockout, knockdown approach in our stem cell derived beta cells. I think the combination and the comparing of those two cell models would be pretty cool as well. But yeah, I think our output would be to try and test the functionality of it and how GCIS is affected.
Samantha, what's next for you? Where you've spoken about defending sometime in the not too distant future, but you know what are your plans?
All right. Well, I am currently both. Well, I've gotten approval to write my thesis recently, so I'm currently writing, but I'm also currently trying to finish up some mouse experiments at the same time. And I have about a month and a half, 2 months before I need to get my thesis to my committee. So I'm on a little bit of a tight deadline, but after my defense, which I think would be in March sometime, I haven't figured out what I'm going to do next. I would like to try and find a lab to do an academic postdoc in for, I don't know, like for however number of years a postdoc is usually nowadays. I know I'm not interested in going to the States, so the US is kind of off the menu for me and my partner for a while. So I'm not going to the States, so I'm planning to stay either in Canada or head over to, I'll try heading over to Europe, I guess, but I don't really know a lot of people there. So yeah, so the answer is I'm searching for postdoc jobs, but I'm also trying to deal with a billion other things to try and complete my degree. It's fun times.
Yeah, that sounds like the end of a PhD to me. So we would, well, we've got plenty of time. So if anyone's got any more questions, please do raise your hand now or put it in the chat. But if nothing comes through in the next few moments, I'd just like to thank you, Samantha, for taking time at this critical point of your PhD to chat to us and be our very first speaker at the EASD Academy Journal Club. And I've certainly really enjoyed learning more about your paper, and I've taken a lot from it. So thank you very much.
Yeah, I mean, thank you for inviting me. This was really chill and unlike any other journal club style that I've had the pleasure to be a part of, but this was really fun. Thank you for inviting me again.
Good. Thank you very much. And so then to everyone else, I'd like to thank you for participating. We'll have another one of these next month, so keep your eye out for announcements about that and what the next paper will be. And in the meantime, we will also have we'll be launching a webinar series in which we will be interviewing individuals, getting feedback, having chats with people. You guys will probably have the chance to interact as well with individuals on specific topics. So our first one will be in November, and again, look out for the date, but that's going to be about science communication and how we can best communicate our research both to the general public and to each other. So keep an eye out for that. And thank you very much.